Assalamu alaikum warahmatullahi ta'ala wabarakatuh. Today we will start new video. If you like it, please like, subscribe and comment to receive more videos in the future. Let's start our video today. Here we go.
بسم الله الرحمن الرحيم والحمد لله وكفى والصلاة والسلام على الحبيب المصطفى رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقه قولي. السلام عليكم ورحمة الله تعالى وبركاته. In this lecture we will talk about the electron transport chain ETC, which is also known as the respiratory chain. This lecture is presented by Elham Omer, MSc Medical Biochemistry, Khartoum University. Let's see the objectives of this lecture first. By the end of this lecture, all the students should be able to define the respiratory chain, identify the general properties of the respiratory chain, discuss the biomedical importance of the respiratory chain, recognize the complexes of the respiratory chain, describe the structure of ATP synthase enzyme, explain the Chemiosmotic theory, differentiate between glycerophosphate shuttle and malate shuttle, and list inhibitors of the respiratory chain. Definition of the respiratory chain. It defines a series of catalysts found in mitochondria that collect and transport reducing equivalents and direct them to their final reaction with oxygen to form water. We mean by catalyst enzymes because the definition of enzyme is protein catalyst. These enzymes found in the inner mitochondrial membrane embedded and found as complexes. We mean by complex enzymes with cofactor. The mission of these enzymes or catalysts is to collect and transport the reducing equivalents in the form of NADH or FADH2 and direct them to their final reaction with oxygen to form water. The electron transport chain is also known as the respiratory chain because the final acceptor of the electrons is oxygen. This figure shows the role of the respiratory chain of mitochondria in conversion of food energy into ATP. Food in the form of fat, carbohydrate and protein after digestion and absorption will be converted into simplest unit. For example, fat is converted into fatty acid and glycerol. Carbohydrate is converted into monosaccharide, mainly glucose, and protein is converted into amino acids. All these are metabolized into acetyl-CoA, which enter Krebs cycle for complete oxidation into carbon dioxide and water. Through this process, there is the production of energy, either in the form of ATP or reducing equivalents in the form of NADH or FADH2. These reducing equivalents are funneled into the respiratory chain for production of ATP. General properties of the respiratory chain. Number one, it is the mitochondrial pathway. That means it occurs in the inner mitochondrial membrane because the enzymes of this chain located in the inner mitochondrial membrane. Number two, it is aerobic pathway. That means it occurs in the presence of oxygen only because the final acceptor of the electrons is oxygen. Occur in tissue having mitochondria only, do not occur in RBCs because RBCs lack a mitochondria and the enzymes of the respiratory chain located in the mitochondria. Biomedical importance. Oxidative phosphorylation enables aerobic organisms to capture a far greater proportion of the available free energy compared with anaerobic organisms. When we talk about glycolysis, under aerobic condition it produces 7 ATP. Under anaerobic condition it produces only 2 ATP. Why? Because under aerobic condition it produces 2 molecules of NADH. Each of them through the respiratory chain produce 2.5 ATP. The total is 5. And there is 2 molecules of ATP produced at substrate level. The total is 7. Under anaerobic condition, these NADH, which produced during the reaction of glycolysis, is used by lactate dehydrogenase to convert pyruvate into lactate, and therefore it has no role in energy production. Oxidative phosphorylation is inhibited by some drugs and poisons. At the end of this lecture, we will talk in details about these inhibitors. The respiratory chain consists of 3 protein complexes: complex 1, complex 3, and complex 4. Succinate dehydrogenase, which actually belongs to tricarboxylic acid cycle, is also assigned at the respiratory chain as complex 2. Also, ATP synthase, which concern with ATP synthesis, consider as complex 5. Therefore, we have 5 complexes. نحن بنقول على complexes because it is enzyme with cofactor. For example, complex 1 contain FMN and iron sulfur protein as coenzyme. FMN refer to flavine mononucleotide. Complex 2 contain FAD as coenzyme and iron sulfur protein. FAD flavine adenine dinucleotide. The complex 3 contain cytochrome B, iron sulfur protein, and cytochrome C1. And complex 4 contain cytochrome A and cytochrome A3. These complexes are embedded in the inner mitochondrial membrane as fixed. We have 2 mobile carriers, ubiquinone or coenzyme Q, which link between complex 1 and complex 3, and also between complex 2 and complex 3. It transfers the electrons from complex 1 and complex 2 into complex 3. Another mobile carrier known as cytochrome C, which link between complex 3 and complex 4. This transfers the electron from complex 3 into complex 4. The nomenclature of the complexes. Complex 1 known as NADH, ubiquinone, oxidoreductase. ال NADH لأن دي reducing equivalence which enter through this complex. Ubiquinone, the mobile carrier, because it link between complex 1 and complex 3. And the enzyme oxidoreductase, which catalyze oxidation reduction reaction. It composed of NADH dehydrogenase enzyme with FMN as cofactor and non-heme iron protein. Complex 2, known as succinate dehydrogenase. It also known as succinate ubiquinone oxidoreductase because it convert succinate into fumarate، عشان كده بنسميه بالسبستريت حقه اللي هو السكسينات. ubiquinone the mobile carrier and oxidoreductase because it oxidize oxidation reduction reaction. It composed of succinate dehydrogenase with FMN as cofactor and non-heme iron protein. Complex 3 ubiquinol cytochrome C oxidoreductase. ده بنسميه بالتو mobile carriers because it receive the electron from complex 1 and complex 2 through ubiquinone، عشان كده بنسميه بالubiquinone. And also it transfer the electron to complex 4 through the mobile carrier cytochrome، عشان كده بنسميه بالتو mobile carrier. It is also known as cytochrome reductase. It composed of heme iron protein known as cytochrome and non-heme iron protein. Complex 4 or known as cytochrome oxidase. It composed of heme iron copper containing protein known as cytochrome AA3, and it has high affinity to oxygen, allowing the respiratory chain to function at maximum rate until tissue become depleted for oxygen. And this is the final complex which deliver the electron to final acceptor oxygen. Components of the respiratory chain. The complexes of the respiratory chain are arranged in order of increasing redox potential. يعني الكومبلكسز دي موجودة embedded in the inner mitochondrial membrane حسب جهد الأكسدة والاختزال بتاعتها. And the electron flow through the chain in step from NAD plus NADH, that means from complex 1 into the final acceptor oxygen. If it pass directly from complex 1 into final acceptor of electron, the oxygen, this will cause thermal denaturation because these electrons has high energy. This energy cause thermal denaturation because these complexes are protein in nature. Therefore, it pass from NAD plus NADH into ubiquinone, then ubiquinone takes this electron to complex 3, and from complex 3 taken by cytochrome C into complex 4, then to the final acceptor of electron, the oxygen. Some substrate with more positive redox potential than NAD plus NADH, like succinate, pass electrons to ubiquinone via complex 2 rather than complex 1. It skips complex 1. The Chemiosmotic theory. This theory explains mechanisms of oxidative phosphorylation. It explains how the oxidation of the components of the respiratory chain lead to phosphorylation of ADP with inorganic phosphates so as to form ATP. This theory postulates that the energy from the oxidation of components in the respiratory chain is coupled to the translocation of hydrogen ions, the protons, from the inside to the outside of the inner mitochondrial membrane. This theory postulates that through the oxidation of the component of the respiratory chain, there will be release of energy. This energy is utilized to pump out the proton from the matrix into outside to the inner intermembrane space. This figure explains the mechanisms of oxidative phosphorylation. Oxidation means addition of oxygen or removal of hydrogen, and phosphorylation means synthesis of ATP. For example, here in the NADH, which comes from Krebs cycle, it undergoes oxidation when it enters the respiratory chain. It loses its hydrogen, which contain electron with negative charge and proton with positive charge. It donates its electron to the components of the respiratory chain. First, it donates this electron to complex 1, and when complex 1 it becomes supercharged, it releases energy. this energy is utilized to pump out the proton into intermembrane space, which is found between inner and outer mitochondrial membrane. Each of the respiratory chain, complex 1, 3 and 4 act as proton pumps، يعني بيشتغلوا كمضخات للبروتونات. complex 1 pump for proton, complex 3 pump for proton, and complex 4 pump to proton through the passage of electron. That means the electron from complex 1 is taken by ubiquinone into complex 3, which pump out 4 proton, then it is taken by cytochrome C into complex 4, which pump out 2 proton. These protons accumulate in the intermembrane space, creating an electrical potential differences between inner and outer membrane. Positive charge outside and negative charge inside, but the inner mitochondrial membrane impermeable for this proton to enter in. Here comes the role of a specific transporter known as ATP synthase. The ATP synthase, which consider as complex 5, spans the membrane and face the matrix. هو عامل زي البودي جارد حارس الممبرين. It has 2 subunits, F0 and F1. This figure shows the structure of ATP synthase. As we said, ATP synthase consists of 2 subunits, F1 and F0. The F1 concern with ATP synthesis, and the F0 or F0 has a cylinder shape. عندها شكل القرص والقرص دوار. When the protons accumulate outside in the intermembrane space, it creates an electrical potential differences between inter and outer membrane. It comes and accumulate around the F0, cause conformational changes and make it to turn. Through this turning, it open and the proton enter in. The protons comes from higher concentration to lower concentration. It has high energy. This energy utilized by F1 to phosphorylate each ADP with inorganic phosphate to form ATP, couple reaction. That means the entry of protons coupled with the synthesis of ATP. The biochemists found that for each 4 proton enter in, 1 ATP molecule produce. Through the oxidation of NADH, 10 proton will be pumped out. 4 from complex 1, another 4 from complex 3, and 2 from complex 4. For each 4 proton enter in, 1 ATP molecule produce. 10 divided by 4 equal 2.5 ATP. So the oxidation of NADH produce 2.5 ATP. FADH2 oxidation lead to pump 6 protons. 4 from complex 3 and 2 from complex 4. Because FADH2 enters the respiratory chain through complex 2. It skips complex 1. And when it enters complex 2, it donates its electron for this complex, which do not pump any proton. Then these electrons is taken by ubiquinone into complex 3, which pump 4 proton, then by cytochrome C into complex 4, which pump 2 proton. For each 4 proton enter in 1 ATP molecule produce 6 divided by 4 equal 1.5 ATP. That's why the FADH2 produce only 1.5 ATP because it skips complex 1 and enter through complex 2. Oxidation of extra mitochondrial NADH. The NADH which comes from glycolysis which occur in the cytosol cannot pass through the inner mitochondrial membrane because the inner mitochondrial membrane has a selective permeability. It permits some substances to pass through it and exclude others. Here comes the role of a specific shuttle known as glycerophosphate shuttle and malate shuttle. In glycerophosphate shuttle, this NADH is oxidized by a reaction catalyzed by glycerol 3-phosphate dehydrogenase, which convert dihydroxyacetone phosphate into glycerol 3-phosphate. In this reaction, the NADH lost its hydrogen and oxidize. This hydrogen is taken by dihydroxyacetone and will be converted into glycerol 3-phosphate. Glycerol 3-phosphate can pass through the inner mitochondrial membrane. عنده حق الفيتو بيقدر يعبر الممبرين. Inside the mitochondria, glycerol 3-phosphate can be converted back into dihydroxyacetone by glycerol 3-phosphate dehydrogenase enzyme. This is the isoenzyme of the cytosolic enzyme. عندي في السيتوسول موجود glycerol 3-phosphate dehydrogenase, and in the mitochondrial the same enzyme. But the mitochondrial enzyme use FAD as coenzyme instead of NADH. And when glycerol 3-phosphate is converted into dihydroxyacetone, it lost its hydrogen. This hydrogen is taken by FAD to form FADH2. And this through the respiratory chain produce only 1.5 ATP. So when the cytosolic NADH enter the mitochondria through this shuttle, it produces only 1.5 ATP. In malate shuttle, NADH, which come from glycolysis, is oxidized by enzyme known as malate dehydrogenase, which convert oxaloacetate into malate. That means the 2 hydrogen from NADH is taken by oxaloacetate to be converted into malate, and the malate can pass through the inner mitochondrial membrane. Inside the mitochondria, the malate will be converted back into oxaloacetate by enzyme known as malate dehydrogenase, the same enzyme, isoenzyme. This enzyme use same coenzyme NAD as coenzyme. This NAD, when malate is converted into oxaloacetate, it releases its hydrogen. This hydrogen is taken by NAD plus to convert it into NADH. And the NADH through the respiratory chain produce 2.5 ATP. So when the cytosolic NADH enter the mitochondria through the glycerophosphate shuttle, it produces only 1.5 ATP. When it enters the mitochondria through the malate shuttle, it produces 2.5 ATP. Inhibitors of the respiratory chain. The inhibitors of the respiratory chain can be classified into inhibitors of the respiratory chain, inhibitors of oxidative phosphorylation, and uncoupler of oxidative phosphorylation. The inhibitor of the respiratory chain are compound prevent the passage of electron by binding to a component of the chain. For example, barbiturate or amobarbital inhibit electron transport via complex 1 by blocking the transfer from FES, the iron sulfur protein, to ubiquinone. Antimycin A and dimercaprol inhibit the respiratory chain at complex 3. The classic poisons H2S and carbon monoxide and cyanide inhibit complex 4 and can therefore totally arrest respiration. Malonate is a competitive inhibitor of complex 2. The antibiotic oligomycin completely block oxidation and phosphorylation by blocking the flow of proton through ATP synthase, and this is considered an inhibitor of oxidative phosphorylation. The uncouplers are substances that allow oxidation to proceed by prevent phosphorylation, are substances that allow oxidation to proceed but prevent phosphorylation. So energy released by electron transport chain will be lost in the form of heat. Example 2,4-dinitrophenol and thermogenin. The thermogenin is a physiological uncoupler found in brown adipose tissue that functions to generate body heat, particularly for newborn and in animal. References for this lecture include Harper's Illustrated Biochemistry, Stryer Biochemistry, Lippincott Illustrated Reviews Biochemistry, Yosemite Kaplan. عندنا more references. عندنا لينينجر ديفلين لو عايزين more details. النت كمان مفيد في بعض الأشياء. عندنا على قناة اليوتيوب في simplified videos ممكن تكون مفيدة. تم الكلام وربنا محمود وله المحامد والعلا والجود، ثم الصلاة على النبي محمد ما ناح قمري وأورق عود. This is all I can say in this lecture. I hope you understood. Thank you for listening and you have a great day.